All right, let's talk about plant reproduction. You know, that topic that seems super easy, but somehow costs students marks every single year. Well, in the next few minutes, we're going to build an exam-proof blueprint to make sure you are not one of them. Let's dive right in.
So, why do students lose these easy marks? I mean, what is it about plant reproduction that trips people up in the exam hall? It's not that the topic itself is that difficult. Nope. The problem is almost always a single point of confusion, a mix-up that almost everyone stumbles over. And you know what? We are going to solve that for good. Right here, right now.
And here it is, the big one, the main culprit. It's the confusion between the layers of the anther wall on one hand and the different types of ovules on the other. I know, they seem kind of similar, and under exam pressure, they just get all jumbled up in your head. But trust me, by the end of this, you will see them as two totally separate, super easy to remember concepts. Promise.
Okay, let's get to it and start building our blueprint. This first section is all about mastering the essential structures. Seriously, think of this as the foundation of a house. If you get this part right, everything else we build on top of it is going to be incredibly simple, so pay close attention. First up, the stamen. This is the male reproductive part of the flower. It's got two main pieces: the filament, which is just the stalk, and the anther, which is where the pollen gets made. Now, you absolutely have to know these two keywords for the anther: Dithecous, which just means it has two lobes, and Tetrasporangiate, meaning it has 4 microsporangia. Write those down. Examiners love these terms. Okay, now let's imagine we're zooming right into a cross-section of that anther. You'd see it's protected by 4 distinct wall layers, and you have to know them in order, from the outside moving in. We've got the epidermis, then the endothecium, then the middle layers, and finally the most internal layer, the tapetum. Getting this order right is absolutely critical for your exam. Look, if you're going to remember just one thing about these layers, please make it this one, the tapetum. Its main job? Providing nutrition to the developing pollen grains. This is, without a doubt, the examiner's favorite layer to ask about. So circle it, star it, highlight it in your notes. Just remember, tapetum equals nutrition. Got it? Okay, quick pop quiz to make sure you've got it. Which anther wall layer provides the precursor for sporal pollinane, that's a key part of the pollen wall, and is the first to break down to provide all that crucial nourishment? Take a second, think about it. Yeah, it has to be the tapetum. See? You got this. So inside all those layers, we find something called sporogenous tissue, and it's packed with microspore mother cells. And listen up, because here is a classic exam trap. It's these mother cells that undergo meiosis to create the pollen grains. Meiosis does not happen in the pollen grain itself. It happens one step before. So many students get that wrong.
All right, let's switch gears and talk about the female reproductive part, the pistil. It's got 3 main components. First, the stigma. Think of it as the landing pad for pollen. Then the style, which is the pathway the pollen tube has to travel down. And finally, the ovary at the base, which holds the precious ovules. It's a simple, logical path. Pollen lands, it travels, and it arrives at its destination.
Now, deep inside that ovary is the ovule, also called the Megasporangium. It has several key parts, like the protective integuments and the embryo sac. But for exam purposes, I want you to really focus on the Micropyle. That tiny little opening is the VIP entrance. It's the entry point for the pollen tube. And this brings us right back to that second major point of confusion, the different types of ovules. The 2 main ones you have to know are Orthotropous, which is totally straight, and Anatropous, which is inverted, kind of like it's bent all the way back on itself. And here's the key fact, that Anatropous, the inverted one, is by far the most common type you'll find in flowering plants. Okay, so you have the core knowledge blueprint down. Fantastic. But now, how do we make sure it actually sticks when you're under exam pressure? Well, this next section is all about locking in that learning with some super simple memory aids and a little real-world context. So here's your first tool. To remember those anther wall layers in perfect order, from the outside in, just remember this phrase. Every exam makes tension. That's epidermis, endothecium, middle layers, and tapetum. See? Simple, effective, and absolutely perfect for a high-pressure situation. And for the main ovule types, we've got another easy mnemonic. Only angry cats. That's O for Orthotropous, A for Anatropous, and C for Campylotropous. And here's a little pro tip to really lock it in. Just remember, A is for almost all plants, because the Anatropous ovule is the one you see most often. So you might be thinking, okay, great for the exam, but why does any of this matter in the real world? Well, this blueprint has massive implications. For instance, if that tapetum fails to do its job, the pollen becomes sterile. And get this, scientists actually use this fact, this male sterility, to create hybrid seeds with all sorts of desirable traits, like better crop yields. The entire field of modern plant breeding is built on understanding these tiny little structures.
All right, you've got the knowledge, you've got the memory tricks, now it's time to put your blueprint to the test. Let's run through some real exam-style questions and see how well you can actually apply what you've just learned.
Okay, I'm about to show you a series of questions. Now, the best way to do this is to actually pause the explainer, solve them yourself, and then we'll go through the answers together. Deal? Ready? Let's go.
Okay, let's start with a classic match the following question. On one side, you've got the parts of the ovule: Micropyle, Funiculus, Chalaza, and integuments. On the other side, you've got their descriptions. Your job is to connect the right part with the right description. Pause here and figure it out. Okay, time to check your answers. How did you do? For the first direct question, the nutritive layer is, of course, B, the tapetum. For the fill-in-the-blank, the most common ovule is C, Anatropous. For the matching question, the correct option was C, which lines up A with 2, B with 1, C with 3, and D with 4. For the multi-statement question, B was the right choice. And for the assertion/reason, the answer was A. Don't worry if you missed one. Just review that specific part of the blueprint.
All right, we are in the home stretch now. This final section is your high score recap. We're going to quickly go over the most common traps and the absolute must-know facts to make sure you walk into that exam feeling totally confident.
Let's build up your defense against the most common mistakes. Here are 3 traps that you are now fully equipped to avoid. Number 1, confusing the nutritive tapetum with the endothecium. Not you, not anymore. 2, forgetting that meiosis happens in the microspore mother cell, not the pollen grain itself. You know that now. And 3, mixing up the micropyle, the entrance, with the chalaza at the opposite end. You're way past that now.
And here we go, your final 30-second rapid-fire revision. Let's do it. The stamen is the anther plus the filament. The anther has 4 wall layers, EEMT. Tapetum means nutrition. Meiosis happens in the microspore mother cell. The most common type of ovule is Anatropous, and the micropyle is the entry point for the pollen tube. Boom! That's the absolute core, right there.
And there you have it. A clear, concise, and totally exam-ready blueprint for the core structures of plant reproduction. You've seen where the confusion happens, and now you have the tools to completely avoid it. So the only question left is this. Now that you have the blueprint, what other complex topic are you going to master next?
But hey, before you click away, how did you do on that little exam test? Drop your score out of 5 in the comments below. I'd love to see how you did. And look, if you're serious about absolutely mastering your exam prep and you want more blueprints just like this one, make sure you subscribe. Until next time, keep learning.